Hello everyone, I'm Trishala Das and I'll be presenting our work on modeling sequence evolution in SARS-CoV-2 spike protein, implications for immune escape and vaccine design. Here is a listing of the people who have contributed to the work and their affiliations can be found below. So to begin with, we are well aware that the evolution of SARS-CoV-2 still presents an ongoing challenge to public health. So understanding the mutation landscape of the viral proteins, particularly those which are responsible for immune escape, is the key to allocate preparedness efforts in advance. Of particular interest, however, is the spike protein because of its ability to attach and fuse into the host cells via the receptor binding domain or the RBD, leading to infection transmission, and also because of it being the primary antigen in the current vaccine designs. So in this study, we built a model that can predict viral protein evolution, which in turn can be used for future vaccine development. We test our model to influenza data and apply it to SARS-CoV-2 using filters for the spike codon sites which are responsible for protein stability and fold, the sites which are responsible for protein function and ACE2 receptor binding, the sites which are important for mutational hotspots, and the sites under immune escape. So to give an overview of the virus evolution, viruses they evolve constantly, undergoing genetic changes leading to new variants and strains that can influence the transmissibility, virulence, host immune response, and vaccine efficacy. So this poses a challenge in predicting and responding to outbreaks impacting global public health. Genomic surveillance has enabled early detection and monitoring of emerging variants, and also much work has been done focusing on the sites under selection pressure using retrospective data. However, methods to predict viral protein evolution that can help in vaccine design is the need of the hour as it can help advance preparation strategies. So this is where the role of deep learning comes in virus evaluation because of its ability to analyze and uncover important patterns from large and dynamic viral datasets, and also because it has the ability to monitor real-time genetic changes, aiding in the prediction of emerging variants. From a technical perspective, forecasting sequence evolution is difficult due to two problems. The first being the combinatorial explosion problem, and the second that there is no assignment of directionality to mutations occurring at a site with time. So in our study, we asked the question that whether we can design deep genetic model which can take care of the above problems and also take into consideration the timestamp or the collection date against each virus sequence. So this is the architecture of a deep generative model, which is a hybrid variational autoencoder model along with the LSTM or the long short-term memory. So the VAE is a probabilistic generative model, and the long short-term memory is a recurrent neural network model which has the ability to capture long-term dependencies in the data, and also which makes it well suited for time series forecasting problems. So in the training part of the data, the discrete inputs are fed onto the encoder, which encodes the data into latent space embeddings. So the latent space embeddings for each sequence are then passed on to the LSTM with the collection date, which gets trained and then is passed through the decoder, which decodes it to the generated output. So the generation part is where the sampler randomly samples from the latent space, passes it on the LSTM, which has the time steps, and then gets passed to the decoder, which constructs the predict sequences. So this is how the VA-LSTM model does. What it does is that given a set of viral protein sequences with the collection date, the preprocessing part takes care of the discrete sequences and converts it into the binary format. The VA encodes the binary format to latent space embeddings or the encoded vector, which are actually continuous space predictions with the collection date. It is then trained using the LSTM and gets decoded back using the VA decoder. Ultimately, we have predicted sequences with the time steps. This can help in future vaccine sequence design. So we have applied our VA-LSTM model to influenza data. So we wanted to see whether our model will be able to predict the influenza vaccine strain for the 2017-18 annual recommendation. So here's a set of filters which we have used to download the data from the NCBI influenza virus database, with the virus type being A, the host selected being human, the country or the region just filtered was northern hemisphere. The protein we selected was the HA for the subtype H3N2. Only full-length sequences and the dates being marked as completed, they were downloaded from the database. So the training set, it comprises of sequences dated prior to 2017, and the testing sequence was the WHO recommended influenza vaccine strain for the year 2017-18 northern hemisphere region for the H3N2 subtype. So here's a listing of the mutations in the training sequences having mutation frequencies greater than 0.1. Rest of the mutations have frequencies less than 0.1 in our data. So the second row shows the mutations which are present in the 2017 vaccine strain. So we wanted to see if our model was able to simulate these mutations in the predicted sequences. So here is the result of the VAE LSTM generated variants of the influenza HA protein being represented by the stem plot, with the x-axis representing the HA protein size and the y-axis represents the mutation frequency of the simulated sequences. The blue vertical line shows the sites which have a mutation present in the simulated sequences, and the green regions highlights the common mutations between the simulated sequences and the 2017 vaccine strain. So here is a listing of the mutations which were actually simulated by the VLST model, and here is a listing of the mutations that were identified as common between simulated sequences and the 2017 vaccine strain. So here we can see that our model was sensitive enough to identify these common mutations but has low specificity. So in the second half of the work, we then applied our model to SARS-CoV-2 spike protein, where the training set is comprised of randomly sampled pre-Omicron sequences coming from alpha, beta, gamma, and delta, along with the reference Wuhan strain, and a testing set comprised of 10,000 Omicron sequences. So here is a listing of the mutations in the training sequences having frequencies greater than 0.1, and the rest of the mutations have frequencies less than 0.1 in our data. So the Omicron specific mutations are marked here, and this is the result of the VAE and STM generated variants of the spike protein, with the x-axis representing the simulated sequences spike codon of the stem plot, and the y-axis represents the mutation frequency. So the blue vertical lines are the uh, ones which are representing the mutations which are simulated. So mutations in the simulated sequences are shown here. So we can see that a total of 286 mutations were simulated, and the common mutations between our Omicron and simulated sequences were also picked out. Particularly, our model was able to identify the mutational hotspot sites as well. So then we wanted to apply some sort of filters which can help us in identifying significant sites. And the first being used as information theoretic measure for predicting fold and function specific residues. So this is an in-house developed pipeline where the relative entropy context is used to identify the fold specific sites and the function specific sites, that is in our case the ACE2 binding sites. So the data we used to identify the sites which are responsible for fold and function is from the Star et al. Paper, where we have used a multiple sequence alignment for the RBD nucleotide sequences for families with ACE2 binding function and without ACE2 binding function. So the basic idea is that the sites which are conserved across both the families is responsible for fold, whereas the sites which are conserved in this family and not conserved in the other is responsible for the ACE2 binding function phenotype. So here is a set of calculations which helps in separation of fold and function specificity. So here is the result of the RE context map, with the x-axis representing the RBD codon sites and the y-axis representing the normalized value of the RE context. So here the upper half of the plot shows the sites which are important for ACE2 binding, and the lower half of the plot shows the sites which are responsible for fold. The second set of filter we used was to identify the hotspot mutation sites, for which we have downloaded data from GISAID, followed by data preprocessing, nucleotide alignment, and annotation. After that, we have assessed the variants and the mutations and plotted their mutation frequencies represented by the stem plot and a heat map. And lastly, we mapped all these mutations and predicted spike epitopes from discotope on the structure. So this is a result of the stem plot representation, where the x-axis represents the spike protein at the nucleotide resolution, and the y-axis represents the mutation frequencies for alpha, beta, gamma, delta, and Omicron. So here we can see a large number of mutations in the Omicron, particularly in the RBD region. So here is a heat map of mutation frequencies for different variants of SARS-CoV-2 plotted against the sites. So here we have on the x-axis the variants of concerns and the y-axis representing the mutations. So we can see that the hotspot mutation site like 614 being picked out. Also we can see the large number of mutations in the Omicron, particularly in the RBD codon site. So this is a result of mapping of mutations onto the structure, where the mutations having high frequencies, they are also accessible on the surface like 614 and so on. And so this was done for all the structures like alpha, beta, gamma, delta, and omicron. The third set of filter we used was to identify the immune escape sites. So actually immune escape is a phenomenon where the host immune system is incapable of responding against an infectious agent, and the process can also be called as immune evasion or antigenic escape. So this occurs during the process of viral evolution, and the mutations which has achieved the process of immune escape are called as immune escape mutations. So say we have a wild type of virus, and the antibodies are able to recognize the epitopes, whereas with the variant with the immune escape mutation, the antibodies fail to do so. So in this result, we have used the structures of SARS-CoV-2, the wild type, and we used the discotope for predicting the B cell epitopes. So here is a result where we can see the x-axis being represented by the spike codon sites and the y-axis representing the discotope score. So this line plot above the default threshold of -3.7 shows certain peaks which are predicted as B cell epitopes. So also if we zoom into the RBD codon region, we can see particular sites being predicted as B cell epitopes also comes in visualization. So we have also used the micron structure to predict the B cell epitopes from discotope, and we can see that the region from 145 to 151 is missing as compared to the wild type. So this region also has a deletion in Omicron from sites 143 to 145, which has been established in the literature as an immune escape mutation, and also has been known to alter antibody recognition as a result of the introduced sequence register shift. So here's a listing of the mutations which are overlapping with the B cell epitopes from our data. And here we can see that when we visualize the mutations on the structure, we can see that the mutations like 440 and 484, these sites are also marked as B cell epitopes from Discuto, and also have high mutation frequencies in them. So lastly, we use all these results together for filtering out mutations in the spike protein for the simulated sequences, particularly in the RBT domain. So the first set of filter we use are from the Discutope result, with the x-axis representing the RBT codon site and the y-axis representing the Discutope score for this particular plot. So here we can see the peaks or the sites identified as B cell epitopes above the default threshold are labeled here, and also we can see interesting patterns via these peaks which are coming up here. So the second set of filter is from a result of Grene et al., where they have identified immune escape sites via deep mutational scanning results across 4 different classes of antibodies. So the x-axis represents the RBD codon sites and the y-axis represents the monoclonal site total escape for that particular site. So here we can see sites like 417, 443 to 448, 460. They have been identified as sites which have high immune escape potential. The third set of filter are the mutations which are present in the Omicron RBD codon site. And lastly, we overlap all these results together onto the simulated sequences. And we can important patterns which are coming up. For example, the site 351 in the simulated sequences has an overlap with the peak in the Discovery plot. The other position can be of 449, which has been identified as a B cell epitope in this group, is also being simulated in our sequence. And the other promising site is the residue numbered 501, which has been identified as a B cell epitope, also has an immune escape potential, and has been identified as a mutation in Omicron, also been simulated by a model. So the other set of filters which we can use as from the RE context map, where we will mask out any mutations which are occurring on the sites which are responsible for fold and function, like 353 and 394, and we can also filter out sites like 520, 523, which are simulated by a model which are also well below the default threshold of a Discotope score and has no immune escape potential, and has shown no mutation frequency in Omicron. So lastly, to summarize our work, we have used generative AI models, LSTM, to predict the sequence of the feature variant. We have implemented analytical methods to predict the map residues which are responsible for mutational hotspots, the sites which are important for protein stability and fold, protein function and ACE2 receptor binding, and the sites which are important for immune escape, to validate and filter mutations in the variant. This protein with the sequence can be used as a vaccine to prevent a future infection. Thank you.